Good evening all of you. You're able to hear me? Good evening all of you. You're able to hear me? Is the audio video clear? Right. Okay, there was a small glitch. Let us start with the session. I hope the audio video is clear. Yes? Okay. So, welcome to Unacademy, the one-stop destination for all English medium UPSC aspirants. And today we are in the second session of Science and Technology Current Affairs. We are in the second session of Science and Technology Current Affairs. And in this class, I will be covering the current affairs from the duration of January to May 2024. This particular session is a part of Conquer Prelims 2025, the crash course for the upcoming prelims. This crash course will happen in 5 phases and this is the first phase, and in that, in the Science and Technology Current Affairs, this is the second lecture. And if you want the annotated PDF of the session, join the Telegram channel where you can find the PDF. You can also get updates about the future sessions in the same channel. Let us continue with the session. The first topic we are discussing is an indigenously made typhoid vaccine. An indigenously made typhoid vaccine. Right. Yes, Shivani, it took me some time to upload that. Okay. Right. So the first topic is about the indigenously made typhoid vaccine. To understand this news article, let us see some basics. First of all, what is an antigen? If you observe here, antigen is any biological substance that can trigger the production of antibodies, right? And if you notice, I said biological substance. I did not say a living material. It can be living or dead, but biological substance. I mean, a plastic cannot be an antigen, or an iron or copper cannot be an antigen. Any biological substance, living or dead, that can trigger the production of antibodies is called as an antigen. Then what is an antibody? Antibody is a defense molecule. It's a defense molecule produced by the immune system. It is a defense molecule produced by the immune system. And antibodies are highly specific to the antigens. Antibodies are highly specific to the antigens. It means the antibody for cholera antigen cannot bind to the polio antigen. Okay, the antibody for cholera antigen will bind only to the cholera antigen, like a lock and key mechanism. They are highly specific. And one more thing, the antibody production is a reaction where the antigen exposure to the antigen is the action and the antibody production is a reaction. I mean, if my body is exposed to an antigen, that is an action. And then my body will produce the antibodies, that is the reaction. It means an antibody can never be produced without the exposure to an antigen. This is a story of an antigen and an antibody. The next term we have to understand is vaccine. All of you answer this question. Let us suppose if we are taking a vaccine for polio, does it contain the polio virus itself or the antibody for the polio? If I take a vaccine for the polio, does it contain the antigen or the antibody for the polio? Yes, the vaccine contains the pathogen itself. Vaccine contains the pathogen itself. It can be a virus or a bacteria or a protozoa, where pathogen means a disease causing substance. The vaccine contains the pathogen itself, and this pathogen is nothing but the antigen. When the antigen enters into our body, then the antibody will be produced, and the antibodies will destroy the antigens. Tell me, what is the function of antibodies? They destroy the antigens. Antibodies will destroy the antigens. So, in one sentence, the principle of a vaccine is something like, okay, manufacturing the weapons. This is something like producing the weapons before the war begins. In one sentence, we can understand vaccines like this: manufacturing your weapons before the war begins. Here the weapons are nothing but, yes, tell me, what are the weapons here? Antigen or antibody? What are we manufacturing as weapons? Very good, antibodies. And before the war begins, the war is nothing but the antigen, that is the pathogen exposure. Okay, so if really a polio virus comes into our body, before we produce antibodies, the virus will start showing its impact on our body. Okay, it means like before we produce antibodies, it can cause all the damage. So the best idea is produce the antibodies before the antigen comes. Like dig the well before we feel thirsty. That is the concept of vaccines. And we have different types of vaccines. Like for example, we have killed vaccines. I mean, take a polio virus, kill it and inject. Because it is killed, it cannot cause polio, but because the structure is intact, it can cause the production of antibodies. Or inactivated. Inactivated. We can also call them as attenuated. Attenuated. Here we don't kill the virus or the pathogen, we just inactivate the virus or the pathogen and inject the inactivated pathogen. Because it is inactive, it cannot cause the infection, but because its structure is intact, it can cause the production of antibodies. Okay. And another type of vaccine is nothing but subunit vaccine. Subunit vaccine. What is a subunit vaccine? Let us suppose this bottle is a pathogen. This is a complete pathogen. In the killed vaccine, attenuated vaccine, maybe killed or inactivated, I am taking the complete pathogen. But if I take a subunit vaccine, I will take only one part of it. I am taking only one part of it. Then we call this as a subunit vaccine. We call it as a subunit vaccine. Right? Okay. We inject only one part of the pathogen. Right. We have many more types of vaccines. Okay. And the types of vaccines we will discuss in some other session. Today's topic is not about the types of vaccines. Just to explain this news article, that is an indigenously developed typhoid vaccine, I have given some background information. Now let us go into the news. Okay, we have seen some background. Let us see about the news article. A Hyderabad-based Bharat Biotech's conjugate typhoid vaccine. Use the word once. What is the type of the vaccine? The type of the vaccine is conjugate vaccine. Okay. Then it is not covered in the types of the vaccines I discussed here. It is not killed. The vaccine made by Bharat Biotech is not killed vaccine. It is not inactivated vaccine. It is not subunit vaccine. It is a conjugate vaccine. Then what is conjugate vaccine? Okay. Conjugate vaccine is, this is nothing but a type of subunit vaccine. Is it clear? Conjugate vaccine is a type of subunit vaccine. Here also we take only one part of the pathogen. But, okay, sometimes what happens, let us suppose this is the typhoid antigen. This is the typhoid antigen. But it is very weak antigen. This is the typhoid antigen, but it is very weak antigen. Weak antigen means what? It fails to trigger the production of antibodies. Antibodies are not producing for it. It cannot trigger the immune system to produce the antibodies. It is a weak antigen. So what we do is, we add another substance to it. We add one more substance to it. This is called as adjuvant. Use the word once, adjuvant. Adjuvant is some structure. When we bind this to a weak antigen, then together the total complex will become a strong antigen. I repeat, conjugate vaccine is a type of a subunit vaccine because we are not taking the complete typhoid bacterium. We are not taking the complete typhoid bacterium. We are taking only one part of it. But the part or the subunit that we are taking is a weak antigen. It is not a strong antigen. Means it cannot trigger the production of antibodies. So we are adding another structure to it. We are calling that particular structure as an adjuvant. The adjuvant plus the typhoid antigen. Adjuvant plus typhoid antigen together forms a strong antigen, and this is nothing but a conjugate vaccine. Conjugate means what? Complex, not a single unit. Combination. Conjugate is something like a combination. It is a combination of the typhoid antigen plus the adjuvant. Together it is called as a conjugate vaccine. Okay, so the news is the Hyderabad based Bharat Biotech manufactured a conjugate typhoid vaccine. Name of the vaccine is Tipbar. Okay, its efficacy lasts for at least 4 years. I mean, if you take the typhoid vaccine today, then it can give protection from typhoid for the next 4 years. Means it is a very efficient vaccine. Currently, there are 2 conjugated typhoid vaccines. The first one is Tipbar, the one that is in the news, about which we are discussing, okay, which is manufactured by the Bharat Biotech, and it received WHO pre-qualification in the year 2017. In the year 2017, this particular Bharat Biotech conjugate vaccine also received a pre-qualification, like a certification. And the other one is Biological E, another company, Biological is, okay, VICRM 197. So the 2 are, one is Tipbar by Bharat Biotech. The other one is VICRM 197. And they both are conjugate vaccines. They both are conjugate vaccines and they both are for typhoid. Right? Okay. And typhoid fever is a bacterial infection. Yesterday also I told you what is a bacteria. Bacteria are unicellular, that is single cell, but prokaryotic. They do not have a nucleus. They are the simplest living organisms, because before that we have viruses, but they cannot be considered as living organisms. So the simplest living organisms are nothing but the bacteria. Okay, typhoid fever is a bacterial infection, not a viral infection, not a protozoal infection. And this is caused by a particular bacterium called as Salmonella typhi. Name the bacteria. Typhoid is caused by a bacterium called Salmonella typhi. And a conjugate vaccine is a type of subunit vaccine. I have written that here also. It is a type of subunit vaccine which combines a weak antigen with a strong antigen called as adjuvant, so that the immune system has a stronger response. That's all. This is the first news article. I hope it is clear for all of you. Salmonella typhi causes typhoid. It's a bacterial infection. Bharat Biotech and Biological E manufactured 2 conjugate vaccines for typhoid. Conjugate vaccines are a type of subunit vaccine where the subunit, when it is a weak antigen, it is added with another substance called as adjuvant. Together it will make the conjugate vaccine. And this tip bar made by Bharat Biotech is a very efficient indigenously developed vaccine. Its efficacy will last for at least 4 years. Okay. Now, moving on to the next topic. Why and how the United States want to establish a time standard for the moon? Okay. What the US is trying to establish? A time standard for the moon. I mean, a standard time for the moon. Okay. As the article itself is saying, why and how. Let us first see why. When we are doing internationally some collaborated efforts, for example ISRO and NASA or European Space Agency and ISRO, okay, like that when 2-3 countries together will do a collaborated effort to reach the moon, and in these 2 countries the times are different, maybe it is 6 p.m. here and 6 a.m. in the other country. So these 2 countries coordinated and working together to send a satellite onto the moon is difficult. So the United States wants to set up a standard time for the moon so that we all can speak in terms of, communicate in terms of the time on the moon. We need not communicate in terms of the time on the earth. So that is the reason why we need to set up a standard time for the moon. I hope the purpose is clear. So we need not communicate Indian time 6 p.m, American time 7 a.m. We need not communicate like that. We can communicate in terms of the time. What is the time on the moon? One standard time. Across the moon, this will be the time. So that is the reason. Okay. Now comes the question, how? How to set a standard time for the moon? To understand how to set a standard time for the moon, we have to understand how did we set a standard time for the earth. Let us first try to understand how did we set a standard time for the earth. Same will be the procedure to set a standard time for the moon. So, how does Earth's time standard works? The coordinated universal time. What is coordinated universal time? This is a standard time on earth. Across the earth, this will be the time. Okay, a standard time on earth is called as coordinated universal time. Be clear with the terms. Okay, yes, Ishwar explaining that. Okay, and all of you, how to set up the standard time is not yet completed. We are doing that. I only told you why. How we are discussing. That is the topic we are discussing, right? Okay. But before we understand how to set up the standard time on the moon, we are understanding how did we set the standard time on the earth. Then same will be the procedure to set up a standard time on the moon as well. So how does earth standard time work? Okay. First, what do we call such standard time on the earth as? We call it as coordinated universal time, UTC. Yes, answer this. What do we call the standard time on the earth? What do we call the standard time on the earth? Coordinated universal time. Okay. And who set this up? Which organization will set up this standard time on the earth? The International Bureau of Weights and Measures in Paris. The International Bureau of Weights and Measures in Paris. I hope it is clear. This is what the standard time is called as. Who set up the standard time on the earth? International Bureau of Weights and Measures in Paris. And what is this? This is an internationally agreed upon standard for the world time. Okay, these are not time zones. This is not Indian time, American time. It is not like that. It is a internationally agreed upon standard time for the complete world, not the universe, world. World means earth. Across the earth, this will be the standard time. Okay, right. Now comes the question, how to set up, okay, the standard time. How to set up the standard time. We set this up using atomic clocks. Use the word all of you. We set up the standard time on the earth. Of course, we set up the standard time on the moon also using the same mechanism. What is that? Atomic clocks. Right? Using atomic clocks. And what is an atomic clock? Okay, listen this carefully. In an atomic clock, 1 second is defined as the time period in which cesium atom, what is cesium? This is one of the elements in the periodic table. Okay, cesium atom vibrates these many times. Is it clear? Okay, we can define 1 second as the time in which cesium atom vibrates. Okay, these many times. Is it clear? Okay, and we can take it as a standard time because the vibration rates of atoms are highly stable. It means like not even one time difference will not come. I mean, this second it vibrated these many times, next second it vibrated just one more time extra or one more time less will never happen. Okay, in 1 second the cesium atom will vibrate exactly these many times. So therefore we define 1 second as a time period in which the cesium atom vibrates these many times. I hope 1 second definition is clear. This is how we calculate the time in the atomic clocks. Why are we calling them as atomic clocks? Because 1 second is defined as the number of times this atom vibrates. Okay, these many times. Is it clear? Right. Now comes the question, why do atoms vibrate? Why do atoms vibrate? Okay, first of all, what is an atom? Look at here, this is an atom. See this complete thing, this is an atom. I hope you are able to see this. This is an atom, okay, and this is the nucleus, and what do we have inside the nucleus? Inside the nucleus we have protons and neutrons, and around the nucleus what we have? We have orbitals. Around the nucleus we have orbitals. In the orbitals, electrons are orbiting around the nucleus. Electrons are orbiting around the nucleus. Is it clear? Okay. So, what causes the vibration here? Okay. So, the movement, okay, or the orbit of electrons cause the atoms to vibrate. Why do the atoms vibrate? Okay, because these electrons are orbiting around the nucleus. This orbit, this movement is the reason why the atoms vibrate. Is it clear? The orbit of the electrons around the nucleus will cause the atom to vibrate. Then do all atoms vibrate at the same rate? I mean, any atom will vibrate same number of times per second? Any atom will vibrate the same number of times per second, or different atoms will vibrate different number of times per second? No. All atoms do not vibrate the same number of times per second. It depends on different atoms have different number of electrons. The number of electrons differ, right? The number of electrons differ. Some atoms will have more electrons, some atoms will have less electrons. Okay. So depending on the number of electrons, how many times an atom orbits around the nucleus, or how many times the electron orbits around the nucleus, or how many times the atom vibrates, changes. So every atom vibrates at a different rate, and we are taking cesium atom as the standard. In an atomic clock, 1 second is equal to. In an atomic clock, 1 second is equal to the number of times the cesium atom vibrates these many times. Okay, right. So let us now go back to this. The standard time on the earth, is tracked by the average of more than 400 atomic clocks. I mean, though the atoms vibrate very accurately, the same number of times every second, still we want to be extra accurate because it is the same time on the complete earth. So it must be super accurate. So we are not depending on a single atomic clock. Is it clear? We are not depending on single atomic clock. We keep 400 atomic clocks at different parts of the globe, in different continents, in different countries. We keep different 400 atomic clocks in different parts of the world. And all the atomic clocks will measure the time. And we take the average of the time of all the 400 atomic clocks. Is it clear? We take average of the time calculated by all the 400 atomic clocks. That is nothing but the standard time on the earth. Is it clear? Right? That is a standard time. Atomic clocks measure the time in terms of resonant, that is vibration frequencies of atoms, such as cesium-133. This is 133rd element in the periodic table. In an atomic time, 1 second is defined as a period in which cesium atom vibrates these many times. As vibration rates of atoms are highly stable, still for extra accuracy we are taking average time calculated by 400 atomic clocks. Okay, and ultra accurate. Atomic clocks makes for an excellent device for gauging the passage of the time. Okay, I hope you understood how the atomic clocks work. That is how we set up the standard time on the earth. Same way we are planning to set up a standard time for the moon. So how to set up a standard time for the moon is clear. Why to set up the standard time for the moon? When different countries work together to launch a lunar mission, then for our communication and coordination, if we speak in terms of earth timings, we all belong to different time zones, so our communication cannot be very accurate. So we should communicate in terms of the moon time. So the same mechanism we can use to set up the standard time for the moon as well. Okay, so that is the news article. Okay, and as we discussed about the universal time on the earth, the standard time on the earth, then how do we calculate the time zones? This part you might have studied in your geography as well. To obtain the local time using atomic clocks, what did we calculate? The standard time across the globe, earth. Okay, now what we are discussing, how to calculate local time. To obtain the local time, countries need to subtract or add certain number of hours from this standard time depending on how many zones they are away from the 0-degree longitude, that is nothing but the Greenwich meridian. If a country lies west to the Greenwich meridian, it has to subtract from the standard time, and if a country is located east from the meridian, it should add some time, a few hours, okay, to that standard time. So this is how the local times are calculated. Standard time is calculated using atomic clocks. Local times are calculated by subtracting or adding a few hours to the standard time. I hope it is clear for all of you. Right. Now, moving on to the third topic in today's session. Okay. Moving on to the third topic in today's session, that is 10,000 Genome Project, Genome India Project. Both mean the same. 10,000 Genome Project or Genome India Project. Okay. This is a news from 27th February from the Hindu newspaper. Let us continue with this. To understand this, we should first know what is the genome. Genome is one set of, not two sets, only one set of all chromosomes in a cell. One set of all chromosomes in a cell, or one copy of total genetic content. One copy of total genetic content of a cell, or one copy of total DNA in one cell. Total DNA in one cell. DNA chromosomes mean the same. DNA, the deoxyribonucleic acid, this is nothing but our genetic material. DNA is nothing but the genetic material, and when the DNA is highly coiled, condensed, tightly packed DNA is nothing but chromosome. Chromosome and DNA are not different. Just a tightly packed, highly condensed form of DNA is nothing but the chromosome. So genome is one set of all the chromosomes, or one copy of the total DNA in one cell. So one genome is equal to, so can you tell me how many chromosomes? One genome is equal to, yes, all of you? Very good. Okay, one genome is equal to 23 chromosomes. Okay. And if one genome is equal to 23 chromosomes, then tell me how many genomes will be present in one cell. In one cell, how many genomes will be present? We have two genomes in one cell, because we have 23 pairs of chromosomes. 23 pairs of chromosomes in one cell. We have 23 pairs of chromosomes in one cell. Out of these two genomes, okay, one genome we get from the mother. I mean, we get 23 chromosomes from the mother, and the other genome, one genome we get from the father. We get one genome from the father. I hope it is clear. We have total 46 chromosomes. In that 23 chromosomes from the mother, 23 chromosomes from the father. Or we have two genomes, one genome from the mother, one genome from the father. So this is about the genome. Okay. As I told you, what is there in the genome? The total genome is made of DNA. The genome is made up of DNA, and DNA is made up of nucleotides. Use the word once, nucleotides. What are nucleotides? Some chemicals, some biochemicals, some chemicals in the living organisms. What are these nucleotides called as? Adenine, thymine, guanine, cytosine. What are they? These are the four nucleotides. What are the nucleotides? Some biochemicals, some chemicals in the living organisms. Okay. Means a DNA will be like this: ATG, CCCT, ATG, CT, AG. Okay. Like this, billions of nucleotides. Okay. The DNA is made up of billions of nucleotides. Is that clear? DNA is made up of billions of nucleotides. Okay. Yeah, ATGCU correct, but we do not have uracil in the DNA. We have uracil only in the RNA. So, in the DNA we have only adenine, thymine, guanine, cytosine. Our DNA may be made up of billions of nucleotides, but all of them are repetitions of just 4 nucleotides. ATGC repetitions. Any number is possible, billions, but just repetitions of 4 nucleotides. This is our DNA. Our genome is made up of this. So tell me, do we all have the same nucleotide sequence? I mean, do we all have the same sequence of ATGC, or we all have a different sequence of ATGC? Is it like we all have the same sequence, or I have GAT-GCC, you have GTA-CCG? We can have different sequences of nucleotides, which is correct. We all have a different sequence of nucleotides, right? All humans have different sequence of nucleotides in the DNA. Okay. Now coming to the next topic, what is genome sequencing? Use the word. What is genome sequencing? Genome sequencing is finding out, okay, the ATGC sequence. I mean finding out the nucleotide sequence of the genome of a person. Is it clear? Okay. I mean knowing whether it is GAT-GCCC or GTA. I mean, what is the first letter? Is it guanine or adenine or thymine or cytosine? What is the second letter? Is it adenine or cytosine or guanine? What is the order or sequence of nucleotides? In which sequence, in which order we have the nucleotides in the genome? Finding it out is called as genome sequencing. Is it clear? Right? So finding out, determining or knowing or reading the nucleotide sequence of the genome is called as genome sequencing. Okay. The first human genome project, first project work in this field is, Human Genome Project. Human Genome Project. This was completed in the year 2003 itself. Okay, not recently. Okay, this project was completed in the year 2003 itself. And you may get one doubt, when we all have a different sequence of nucleotides in the genome, so whose sequence did they genome? Okay, whose sequence did they find out in this project? When we all have a different genome sequence, whose genome did they sequence in this project work? Not one person, a few lakhs of people, okay, few lakhs of people from across the world, Is it clear? Okay. Yes, Lalita Prasad, genome sequence, not genome sequence, genome sequencing means finding out the sequence of letters, finding out the order of the letters, in which order the nucleotides are placed in my genome. That mechanism, finding it out, is called genome sequencing. Okay, genome sequencing. Okay, but because we all have a different sequence of nucleotides in the genome, they did not sequence the genome of only one person. I mean, they did not try to find out the nucleotide sequence of a single person. They sequenced the genomes of a few lakhs of people. And this sample is taken from where? Only from one country or from across the world? Everybody please. These lakhs of people are taken from all over the world. This is the first human genome project completed in the year 2003. Means it is an international project. But, okay, not many countries, all countries, I mean all over the world, from across the world, from different countries, they have taken the few lakhs of people and collected their DNAs and try to read the order of nucleotides in their DNAs. That's it. Okay, I repeat, from all over the world, they have taken a few lakhs of people, then extracted their DNAs, and then they tried to read or identify the order of nucleotides in their DNAs. This is the first such project work called Human Genome Project, completed in the year 2003. This was an international project, and all this is some static content. Now let us see about the news. What is there in the news article? Okay. This is 10,000 genome, not lakhs of people. 10,000 genomes, only 10,000 people's genome sequenced. I mean, we tried to read the nucleotide sequence of only 10,000 people. And that is also called Genome India project. So from the name, tell me, is it an international project or all the 10,000 people are taken only from India? All the 10,000 people are taken only from India. Okay. The Department of Biotechnology announced the completion of this 10,000 genome project. It means they completed reading the nucleotide sequence of all the 10,000 genomes. They completed reading or finding out the order of nucleotides in all the 10,000 genomes. That is the news, right? Okay. This is an attempt to create a reference database of whole genome sequence out of the country. And United Kingdom, China, US are those countries which have also sequenced approximately 1 lakh of their genomes. I mean, UK sequenced 1 lakh genomes of their citizens. China read the nucleotide sequence of 1 lakh genomes of their citizens. Like that, these are a few countries, okay, in which also similar project works, okay, took place. About 20 institutions across the country were involved in this project. This project was not completed, okay, by a single institute. Around 20 institutes took part in this particular project work, with Indian Institute of Science in Bangalore and Center for Cellular and Molecular Biology in Hyderabad being the lead institutions, and 18 more institutions cooperated in this process. The lead institutes are, okay, CCMB Hyderabad and IISc Bangalore. Is it clear? Yes, covering all the ethnic groups, covering different races. Is it clear? Right. So this is about the completion of 10,000 genome project. Okay, Genome India project. That was the news. Now moving on to the next topic. Okay, and stay more focused. We are going to discuss after this. Okay, after this we are going to discuss a very important topic about the Higgs boson, how the matter is created, how the matter got its weight or mass, the neutrinos, neutrino observatories, Large Hadron Colliders. These are some very important all time. They are not only 1.5 year back current affairs, they can also be considered as classical topics, always important. Okay, so we are going to discuss them, but before that we are going to discuss 2 more topics. Okay, and the fourth topic in today's class, okay, the fourth topic in today's class is the discovery of new cellulose digesting bacteria. Okay, tell me, what is the speciality of this bacteria? This bacteria can digest the cellulose. Okay, right. To understand this, we should first know what is a cellulose. Cellulose is present in the cell wall of plant cells. Is it clear? Animals do not have a cell wall, so animals do not have cellulose. So if you are a non-vegetarian, if you consume primarily meat, then we consume very little amount of cellulose. But if you are a vegan or a vegetarian, and when you consume more plant-based diet, then you eat a lot of cellulose. Particularly, it is rich in the fiber-rich food. It is present mostly in the fiber-rich food, right? Clear till now? That is cellulose. Originally, we have an organ called as appendix. Name this organ once. We have an organ called as appendix. Appendix produce an enzyme called as cellulase. Enzyme called as cellulase. Don't confuse between cellulose and cellulase. Focus on the end part of the word. Lase. Cellulose is a substance. Cellulose is an enzyme that will digest the cellulose. And what is an enzyme? Enzyme is nothing but a biocatalyst. Enzyme is nothing but a catalyst, something which will speed up the reaction. When a catalyst is in the living organisms, we call it as enzyme, a biocatalyst. Is it clear? The name of the enzyme is cellulase. Which organ produces this enzyme? Appendix. But, okay, because the humans became omnivores, humans during the evolution became omnivores, started consuming the meat as well, not only depending on the plant food just like our ancestors monkeys. Okay, we also eat the meat. Okay, omnivores. Plus also we started consuming processed food. Means instead of eating the whole fruit, we are preferring to drink a fruit juice. And I told you cellulose is rich in the fiber-rich food. Instead of a whole fruit, when you take a fruit juice, you will not get enough cellulose. There is no cellulose. Or we are peeling the skin of the vegetables or fruits and then we are consuming. So it means we started consuming very little cellulose. Okay, so because of this, what happened? Cellulose consumption decreased. Clear for all of you? Cellulose consumption decreased. Because cellulose consumption decreased, appendix became a, can you fill the blank? Dash organ. Appendix became a, yes, vestigial organ. Vestigial organ means what? Okay, not working. It is there but not working. We have appendix in our bodies, and sometimes some people will get appendicitis also. That is an inflammation to the organ appendix. Okay, so we have appendix but it is not working. It is not producing cellulase. So now tell me, all of a sudden today if I eat some sprouts or some fiber rich food, do I have cellulase enzyme to digest it? All of a sudden if I eat some fiber rich food, do I have cellulase enzyme to digest it? Yes? No, we don't have. Because the appendix became vestigial organ. The appendix became vestigial organ, not producing the cellulase. Then comes the question, then how are we digesting that? Okay, when I eat sprouts or sometimes when I eat a whole fruit, how am I digesting it? Okay. In some earlier discussion also, if you remember, I told you that we have some friendly bacteria in our body, isn't it? We have some friendly bacteria in our body, right? Okay. They are mainly present in the gut. Gut means what? Our digestive system. They are mainly present in our gut, digestive system. Okay. We have all over the body, like say for example, we have on the hands, on the skin, on the hair, in the mouth, everywhere. We have these friendly bacteria everywhere in our body. And surprisingly, in our body we have more bacterial cells than our own cells. I mean, I have more bacterial cells in my body than my own body cells. Those many friendly bacteria we have in our bodies, they perform so many functions. They help in digesting the food. They also produce vitamins for us, okay, and they also decide our moods. Are you a happy person or a sad person? So like that, countless, okay, and more research is coming about this friendly bacteria, and we are coming to know that they are helping us in countless number of different ways. And one way is producing the cellulase enzyme, a new cellulase producing, means cellulose digesting bacteria. One of our friendly gut bacteria is digesting the cellulose. Why? Because I cannot produce cellulase because my appendix became the vestigial organ. I repeat, I cannot produce cellulase enzyme to digest the cellulose because my appendix became a vestigial organ. But still we are digesting the plant food. Sometimes we eat fiber-rich food and we are happily digesting it. How is it happening? From where we are getting this cellulase enzyme? Our friendly bacteria in the digestive system, gut bacteria, are producing it. Is it clear? The study found 3 new species of cellulose digesting bacteria. Cellulose digesting bacteria means cellulase producing bacteria, because we need cellulase to digest the cellulose. These bacteria are found prevalent in great apes, that is chimpanzee, gorilla, etc. Ancient human societies like some Neanderthal man, early human beings, okay, and also hunter-gatherer communities. Why these? Because they consume more plant food, isn't it? Chimpanzees, great apes, other gorillas, orangutans, such animals, and also the ancient humans or primitive tribal groups like hunter-gatherer communities, they depend more on the plant food. So they need more cellulase enzyme. So in their gut, in their digestive system, the scientists found 3 new species of the cellulose digesting bacteria. Is it clear? Okay. The importance of cellulose digestion. Cellulose is the main component of the plant fiber which nurtures the gut microbiome. Okay. Humans rely on the gut microbiome to digest the cellulose, as cellulase producing organ appendix became the vestigial organ. Because our appendix is not working, we depend only on this gut microbiome, that is gut bacteria, for the production of cellulase. Newly discovered bacteria convert cellulose into short chain fatty acids. Okay, they convert the cellulose, they digest it and convert it into short chain fatty acids. Okay, so that is the news. Moving on to the 5th topic in today's class. Okay, this is about the newly discovered organelle that help in nitrogen fixation. Okay, a newly discovered organelle that helps in nitrogen fixation. Okay, so for this Lalita Prasad, the bacterial species were not mentioned in the news. So it's not important to know the species of the bacteria. There are so many species of the gut microbiome. Okay, and the main purpose of this article is to know an additional function performed by the gut microbiome. Like there was one of the prelims previous question, okay, about the gut microbiome only, what all they can do. They have given the functions of the gut microbiome and the statements, that is produce the vitamins, digest the food, like that 3-4 statements are given. Okay, and they asked which one is the function of the gut microbiome. Like that they can also include the digestion of cellulose. That's all. This is another function performed by the gut microbiome. That's it. The species were not mentioned. That is not very important to remember as well. Okay. Why appendix became vestigial? Because we stopped using it. Why did we stop using it? Because we stopped consuming lot of cellulose. Okay, I explained all that here. Because we started consuming processed food, so the fiber intake decreased. We started consuming the meat, So the plant food intake decreased. We did not use the appendix, so it became a vestigial organ. So we are depending on the gut microbiome to digest the cellulose. Clear? Okay. Now, moving on to the next topic, the fifth topic in today's class. Okay. Can the newly discovered organelle help the plants in nitrogen fixation? Okay. To understand this also, we need to know some background information. That is, okay, what are the organelles? Use the word once. What are the organelles? These are the cellular level, I mean miniature organs in a cell. Miniature organs in a cell. Okay. Like, did you all hear about mitochondria? Mitochondria performs respiration. Okay. Mitochondria can be considered as the lungs of the cell. I mean, at the body level, if the respiration is done by lungs, at the cellular level, the respiration is performed by the mitochondria. Is that clear? Yes, we call it as powerhouse of the cell. Okay. And we have a nucleus. It can be compared to the brain of a cell. It means just like we have brain in our body which coordinates all functions in the cell, same way we have nucleus in the cell. So what are the organelles? Organelles are the small miniature organs or cellular level organs. Okay. We have them like this. I mean, if this is the cell, inside the cell you have some liquid or a fluid called as cytoplasm. And inside the cytoplasm you have these organelles. Like, for example, this is the mitochondria, nucleus, like that. I hope it is clear. So this is about an organelle. And the next topic we have to understand is what is nitrogen fixation. Nitrogen fixation means the molecular nitrogen, that is N2. N2 is N triple bond 2. This is how we have nitrogen in the atmosphere. Is it clear? In which form we have nitrogen in the atmosphere? N2. N triple bond N. And this is a very strong bond. Means very difficult to break. Is it clear? It is not easy to break this bond. Clear for all of you? Okay. So therefore, plants and animals, both plants and animals cannot use the molecular nitrogen. We have enough nitrogen in the atmosphere. All around us we have enough nitrogen in the atmosphere. But unfortunately, we cannot use it. This is just like a sailor feeling thirsty in the middle of an ocean. The ocean is full of water, but the sailor cannot drink that water. Okay. So like that, we have enough nitrogen, but we cannot use it because we cannot break this particular bond. Yes, clear for all of you? So, okay, till now what we know is only some bacteria can do it. Plants and animals cannot do it, but only some bacteria can do it. Is it clear? Okay. Now, let us go into the news and discuss a few points and then again come back to the background knowledge. Scientists recently found an organelle performing nitrogen fixation. It means, okay, look at here, just like the mitochondria performing respiration, just like the chloroplast just like the chloroplast performing photosynthesis. Same way, they found a new organelle. Okay, this is a new organelle. Means till now we don't know that one such organelle exists. We did not study about this in the NCERTs. In the NCERTs, in your school days, you studied about mitochondria, chloroplast, nucleus, you know all these things, but you never heard about an organelle that can fix the nitrogen. Okay, so this is a new organelle, newly discovered organelle that can perform nitrogen fixation. Then what is nitrogen fixation? Okay, converting nitrogen into ammonia or nitrate or nitrite. Is it clear? That is usable forms of the nitrogen. Okay, what are the three forms in which plants or animals can use the nitrogen? Okay, ammonia, nitrates, nitrites we can use, but molecular nitrogen N2 we cannot use because of that strong triple bond between the two nitrogens. Okay, normally bacteria can convert this nitrogen into, okay, what can convert this nitrogen into ammonia, nitrates and nitrites, etc. Only bacteria can do. This is what is our old knowledge. This is what we know till now. But what is given in the news recently? Okay, the news is they found a new organelle. That can perform the nitrogen fixation. Just like mitochondria can perform respiration, this organelle can convert nitrogen into ammonia or nitrite or nitrate. Okay, so all of you tell me, what is this newly discovered organelle doing? Converting the N2 into ammonia or nitrite or nitrate. Okay, I mean fixing the nitrogen or performing the nitrogen fixation. Yes, clear for all of you? Right. Okay, so scientists recently found an organelle performing nitrogen fixation. This organelle could be engineered into the crop plants. So now the idea is, take out this organelle, keep it in your wheat plant cells. Then what happens? The plant can perform the nitrogen fixation. I hope you understood this. Then this wheat plant can fix the nitrogen. I hope it is clear. Just cut paste. Okay, cut this. I mean take this organelle from the cell in which it is present. Transfer it to, okay, cut from here and paste it in the wheat plant cells or rice plant cells or apple plant cells or any plant cells. Is it clear? Then the plant cells can fix the nitrogen. I hope it is clear for all of you. So this is nothing but an engineered plant. This is not a natural plant. Can a natural plant fix the nitrogen? I mean, can a natural plant convert the molecular nitrogen into nitrate or nitrite or ammonia? Can a natural plant fix the nitrogen? No. Okay. No, a natural plant cannot fix the nitrogen, right? But, okay, into this plant what we have done? We transferred this nitrogen fixing organelle. So now this plant can perform the nitrogen fixation. This is an engineered plant. Okay. No, there is no name for it. Okay. And some of you are asking me, is it a genetically modified crop? Is this a GM crop? Good question. Is it a GM crop? You guess, is it a GM crop? Can we call it as genetically modified crops? Very good guess, very good guess. This cannot be called as genetically modified crop because we did not transfer a gene into the wheat plant. If you transfer a foreign gene into the wheat plant, then we call it as genetically modified crop. But what did we transfer here? Not the DNA, not the genetic material. We transferred an organelle. Okay, so this is a new technique. So I just called it as an engineered plant, but I did not call it as a genetically engineered plant. Is it clear? No, this is not genetical engineering, Rohit. This is not related to genetic engineering. Okay, because simple logic. Did we transfer the gene? No. Okay, fine. Let us see a little bit more clarity on this. Where do we have the genes? Okay, here we have the genes, right? This is the nucleus. This is the nucleus. In that we have the DNA. In that we have the genes. If I transfer genes into a plant, then we call the plant as genetically modified or GM crop or genetically engineered. This is an engineered plant but not genetically engineered. I hope it is clear because we transferred an organelle, okay? Not the DNA, not the gene, okay? Now tell me, can this engineered plant fix the nitrogen? Can this engineered plant fix the nitrogen, convert the nitrogen into ammonia or nitrates or nitrites? Yes, all of you? Yes, very good. Nitrogen is a key component in the proteins and DNA of all the living organisms. Why nitrogen fixation is important? Where do we use nitrogen? We need nitrogen in our proteins, in our DNA, etc. Nucleotides are made up of nitrogen in the DNA. Amino acids in the proteins are made up of nitrogen. So we need nitrogen in our DNA, proteins, etc. So nitrogen is very important for us, but we cannot fix it. We and plants also cannot fix it. But by transferring this organelle, okay, we can make the plant capable of fixing the nitrogen. I hope it is clear. Yeah, legumes can fix the nitrogen, but not directly, through some bacteria in the nodules of their roots. So no plant can fix the nitrogen. Legumes can fix, but legumes are not fixing. They are fixing the nitrogen with the help of some bacteria present in their nodules. So yes, the statement is correct. Plants and animals cannot fix the nitrogen, but this plant can fix the nitrogen because we transferred a newly discovered nitrogen fixing organelle into this plant. But this is a hypothetical technique. This is a plan, future plan. They have not done yet. Okay, they did not transfer this organelle into a plan yet. That is a future plan. So what is the news? News is only they found this new organelle. They are planning what all we can do with this new organelle. That's it. Is it clear? No plants, Srinivas. That is only a plan. They are planning. Okay, if we transfer like this, the plants can produce the nitrogen. The plants can fix the nitrogen. That is just an idea. They did not transfer it. Clear? Then, although nitrogen gas makes up 78% of the Earth's atmosphere by volume, plants and animals lack a system that can utilize the atmospheric nitrogen. I hope it is clear. Okay. And bacteria and archaea, I already told you this, I am just giving it a reading. Only bacteria and archaea can convert atmospheric nitrogen gas into ammonia by nitrogen fixation. Unlike many free-living nitrogen-fixing bacteria, legumes are a class of plants in the family called Fabaceae. Legumes belong to a family called Fabaceae. They bear the nitrogen-fixing bacteria. As I just told you, legumes will not fix the nitrogen. They harbor a type of bacteria, the nitrogen-fixing bacteria, in their root nodules. Okay, like this, their roots will have nodules. These nodules will have bacteria. They will fix the nitrogen. Okay. The origin of new organelle performing nitrogen fixation could be explained by endosymbiotic theory. Okay. Use this word once. Endosymbiotic theory. Use this word once. You all know the meaning of the word symbiosis, right? What is symbiosis? Symbiosis is nothing but mutual help, right? Then endo means what? Inside. Okay. So what happened is, let us suppose there are two cells. Listen carefully. What I am explaining now is the origin of this new organelle. The origin of this new organelle. How this new organelle is originated. Is it clear? Endosymbiotic theory explains the origin of this new organelle. Endosymbiotic theory explains the origin of this new organelle. To explain endosymbiotic theory, I am clarifying certain terms. Symbiosis means mutual help. Endo means inside. So what am I explaining now? Endosymbiotic theory. Why am I explaining endosymbiotic theory? To explain the origin of this organelle. So let us try to understand this. This is, okay, a cell called A. Okay. Then this is another cell called B. This is another cell called B. Okay. Right. I'll just draw it a little bit small in size. Okay. This is another cell called as B. Okay, this is a cell, this is a cell. Is it clear? What are A and B, all of you? What are A and B? They are two different cells. Now what happened? A eats away the B. Did you understand? A eats B. Now the B is inside the A. Did you understand? Now the B is inside the A. Got it? Okay. Now this B cell was capable of performing nitrogen fixation. The cell B can perform nitrogen fixation. Is it clear? Cell A cannot perform nitrogen fixation. Cell A cannot perform nitrogen fixation. Clear? Right? I repeat, there are two cells. Cell A cannot perform the nitrogen fixation. Cell B can perform the nitrogen fixation. Point number 3. Cell A eats away the cell B. Right? Now the cell B is inside the first cell. Is it clear? This is inside the first cell. Now A eats the B, and after that, okay, B became an organelle inside the cell A. I hope you understood the endosymbiotic theory. I repeat, there are two cells. The first cell A cannot perform nitrogen fixation. The other cell B can perform nitrogen fixation. Then the cell A eats away the cell B. Now the cell B is inside the cell A. Once this cell is inside the cell, once the cell B is inside the cell A, now this B became an organelle inside the cell A. After A eats away B, B became an organelle in the A. A is using B as an organelle, and this organelle now can perform. Okay, so this became a new organelle that can perform, the nitrogen fixation. Is it clear? Okay, it will have DNA. Such organelles. Good question, Aditya. Such organelles. Not only this, you know mitochondria. You can note down the points. Mitochondria, chloroplast, and this new organelle, okay, this new organelle, there is no name for it, that fix the nitrogen, okay. All these are originated by the same mechanism, endosymbiosis. Endosymbiosis. Is it clear? Okay, I explained endosymbiosis, okay, to explain the origin of this new organelle. Is it clear? Endosymbiotic theory explains the formation of this new organelle. But interestingly, the same theory explains the origin of mitochondria, chloroplast also. I mean, okay, this is a big cell. There are 3 small cells. This cell can perform respiration. This cell can perform photosynthesis. This cell can perform nitrogen fixation. I hope you understood. This is A, this is B, C, D. Then A eats away B, C, D, all of them. Okay. B became mitochondria. Okay. And C became chloroplast. D became this new organelle. I hope it is clear. That is the reason why these 3 organelles have their own DNA. These 3 organelles, the 3 organelles have their own DNA. Some of you might have heard that, okay, mitochondria also will have some DNA. This is the reason why mitochondria will have some DNA, because it is formed by endosymbiotic theory, right? So this is a theory explaining the origin of mitochondria, chloroplast and the new organelle. Is it clear? Yes, A is a very hungry cell. Okay, can we continue? Right. Now moving on to the next topic, the most awaited topic, that is Large Hadron Collider, Higgs bosons, Higgs field, etc. Let us try to understand about it. Okay, right. To understand about Higgs boson, Higgs field, Large Hadron Colliders, neutrino observatories. Did you hear these words? Neutrino observatories, Large Hadron Collider, Higgs boson, the God particle, Higgs field, etc. Let us try to understand all these terms and then we will go into the news, right? Can we proceed? Right. So, now we are discussing, okay, yes, I am explaining from the base. Azhar, I will explain every topic from the base. Like when I discussed the genome project, I started from what is nucleotide, what is genome. I did not just explain only the news. After 5-10 minutes, we are going into the news. Okay. Same way I am explaining from absolute basics. Wonderful topic, complete focus please. Okay. This is the structure of an atom. Look at here, because I didn't want to waste your time. I have drawn the diagrams beforehand. Okay. So let us try to understand the structure of atom. Okay. All of you tell me, what is this? What is this? Keep answering. What is this? This is an atom, right? This is an atom. Okay. What is there inside the atom? What is this? What is there inside the atom? Right? Nucleus. What is there inside the atom? Nucleus. Right. What are there inside the nucleus? What are there inside the nucleus? You know, proton and neutron. Proton and neutron. Listen carefully and don't say it is difficult. This is atomic structure. 8th class, 9th class students read this. Just more focus please. Okay. So atom, nucleus, inside the nucleus protons and neutrons. Till this point you also know, you studied in your school days, right? But in general, we consider electron, proton, neutron as the smallest particles, but no, that is not correct. Okay, the proton, look at here, the proton is made up of quarks. Can you see some yellow color balls here? The protons are made up of quarks. We have two types of quarks. Up quarks, up. Okay, I did not write the word up, but I put upward arrows. Up quarks, down quarks. Is it clear? Quarks are two types, up quarks, down quarks. Okay, a proton will have two up and one down. I mean up, up, down, then it is proton. Down, down, up, then it is neutron. I hope you understood. This is neutron. This is also made up of quarks. But the quarks in the neutron are down, down, up. I hope you understood the difference between proton and the neutron, right? So protons and neutrons are not the smallest particles. Inside the proton also we have quarks. I hope it is clear. Then, okay. Can you see some red color zigzag structure here between the quarks? That is called gluons, glue, like a fevicol, glue, gum. Okay, it is like a gum that is holding the quarks together. Okay, a gum, like not literally gum, it is like a force. Okay, it is a force which is holding them together. Is it clear? Okay, that is called gluons. So quarks bound by gluons, quarks bound by gluons. I'll show you the photograph also. Look at here. Let us imagine, okay, this as the atom. In the atom, this is the nucleus. In the nucleus, there is one proton. Proton will have up quark, down quark, and gluons are the forces that are holding the quarks together. I hope it is clear. No, that is not a wire. It is drawn like that. Okay, right. More focus, please. This is a difficult topic, deeper topic, right? Clear till now? But where are electrons? Electrons are not inside the nucleus. Electrons are not inside the nucleus. They are orbiting around the nucleus. In an earlier diagram also we have seen they orbit around the nucleus in the orbitals. Is it clear? Electrons are here. And electrons are smallest particles. Okay, there is nothing inside the electron, and according to our present knowledge, I mean in future we may find something inside the electron also. At the moment we don't know about anything which is present inside the electron. I hope it is clear. Okay, so atom, nucleus, proton, neutron, electrons, gluons, clear. Now, okay, read this word. Use the word once. What is this? Hadrons. This is nothing but the hadron in the Large Hadron Collider. Okay, LHC, you might have studied about, heard about LHC in some news earlier. Large Hadron Collider, hadron. What are hadrons? Okay, hadrons are subatomic particles. Hadrons are some subatomic particles. Clear? Hadrons are subatomic particles. I mean particles present inside the atom. Clear? Yes. Okay. And hadrons are two types. Hadrons are two types. Baryons, mesons. Use both the words. Hadrons are two types. Baryons, mesons. Baryons, the word you can easily remember, bary, bhari. I mean these are the heavier ones. Okay, these are comparatively heavier ones. Clear? Therefore, they are called as baryons. Okay, you can remember that easily as bari. I repeat, hadrons are the subatomic particles. There are two types, baryons and mesons. Baryons are again two types, protons and neutrons. I told you that they are heavier. Electrons are lighter, isn't it? Electrons are lighter. They don't have anything inside them. But whereas protons and neutrons, they have quarks, gluons, all those things. They are heavier ones. Okay, so baryons examples are protons and neutrons. Is it clear? And same way, mesons are two types, charged mesons, uncharged mesons. Keep using the words and keep memorizing the words. Actually, we need a little memory also to understand the topic. In general, we keep wondering between should I understand or should I mug up. In my opinion, they both are not two different mechanisms. We need both of them. Like, for example, if you forget the character names in a novel, then you cannot understand the story in the novel. Okay, so if you forget these words, you cannot further understand what we are discussing about. So keep memorizing the terms, understanding the concepts. So we need not worry whether we have to mug up or understand. We have to do both. Mug up the terms, understand the concepts. As simple as that. Yes or no? Okay, right. Okay, so baryons and mesons. What are the baryons? Protons and neutrons. Is it clear? Okay. Monu Chowdhury, before that, remember that this is 8th class topic, and if 8th class topic is going from above the head, then like which level we have to discuss. So this is atomic structure, right? Okay, I hope it is clear. So hadrons are subatomic. For you, Monu, I am repeating once again. Hadrons are subatomic particles, particles present inside the atom. There are two types, baryons, heavier ones, baryons. And the other type, little lighter ones, are mesons. Okay, and baryons are again two. What are they? Protons, neutrons. See, this proton, neutron are baryons. You can easily remember it. How do you know that they are the heavier ones? Because they have again this quarks and all those things in this. Is it clear? Clear till now? Whereas mesons are little lighter ones. Mesons are little lighter ones, right? Then mesons are again two types. Okay, what are they? Mesons are again two types. Okay, charged mesons and uncharged mesons. Okay, and mesons are very unstable. Mesons are very unstable. Okay, like very quickly means in 1 hour, 1 minute, 1 second, 1 millisecond, no. In 1/100th of a millisecond, in 1/100th of a millisecond, the mesons will get converted into, okay, mesons will get converted into electrons, neutrinos and photons. Is not neutrons, neutrinos. Okay, don't get confused. Neutrons are baryons, neutrinos are mesons. Is it clear? Clear till this point? Mesons are two types, charged mesons, uncharged mesons. And mesons are highly unstable. Very quickly means 1/100th of a millisecond, not even 1/100th of a second. Okay, in that much small time, the mesons will get converted into, if it is a charged meson, it will get converted into electron and neutrinos. If it is an uncharged meson, it will get converted into photons. Photons are, you know, packets of light, we call packets of energy, isn't it? We call photon as a packet of energy. Is it clear? Right. Okay. So simple, I'll just repeat once again. Hadrons are the subatomic particles, particle inside the atom. There are two types, baryons and mesons. Baryons are heavier ones, mesons are lighter ones. Baryons are two types, protons and neutrons. Okay, and mesons are two types. They are uncharged mesons and charged mesons. And mesons are very unstable, very quickly they get converted into the other particles. And if it is a charged meson, it will get converted into either electron or neutrino, not neutrons, neutrinos. Okay, and uncharged mesons will get converted into photons. Okay, these are all the subatomic particles. This protons and neutrons, they are baryons. Okay, like this, let us write here. These are baryons, these are mesons. Got the complete clarity? So this is the atomic structure. Gluon is a force carrier. It is a force holding the quarks together. Okay, gluon is nothing but the force which is holding the quarks together. I hope the atomic structure is clear. Okay, now let us go back into the right, next topic is, what is neutrino? What is neutrino? It is a meson. What is meson? It is a subatomic particle. But yes, forgot to tell you one very important point. Okay, these neutrinos and photons, they are not part of an atom. They are present free. They are not inside the atoms. Is it clear? Electrons, protons, neutrons are present in the atom. But neutrinos and photons, they are not neutrinos and photons. They are not a part of the atom. They are present free, not inside the atom. Is that clear? Electrons, protons, neutrons are inside the atom. So I have shown them in this atomic structure. But neutrinos and photons, I did not show them inside the atom because they are not in the atom. They are present free. Okay, and okay, so these are the fundamental particles. I hope it is clear till now. Okay, news you cannot directly understand, Ishwar. We have a lot of story behind the news. And also they will never ask a question on the news. I mean, did you ever see in UPSC, okay, which among the following article is published in the Hindu newspaper 22nd January 2024? Did you any time see such question in any UPSC exam? Yes. Okay. They will never ask a question from the news. When there is a news, they will ask questions from the topics covered in the news. Right? Okay. Then I hope these particles are clear. So next we are discussing, okay, what is Higgs field? What is Higgs field? Look at here, Higgs field. This is news. Okay, this is news. Happy? Right. So now let us try to understand what is Higgs field. Okay, just after the Big Bang. Okay, you know Big Bang, the origin of the universe. Okay. Just after the Big Bang, Higgs field was zero. Okay, there is no nothing called Higgs field soon after the Big Bang. Is that clear? But as the universe cooled and the temperature fell below critical value, after some time what happened? The temperature decreased below certain point. Then, okay, spontaneously a field got generated. A field grew spontaneously. Are you all following clearly? A field grew spontaneously, right? And I know that the red is not very brightly visible on this slide and we have only a few color options. And when I want to maintain a contrast, I am using these colors, right? Most of the things I am explaining in the lighter colors only, right? So now coming back to this, okay. Higgs field grew spontaneously after some time. Okay, I repeat, Big Bang occurred. The moment the Big Bang occurred, there is nothing called Higgs field. After some time, the temperature reduced. Once the temperature reduced spontaneously, okay, a field called Higgs field formed. Okay, how did it form? Nobody knows. Is it clear? That is not you or me, nobody knows. But spontaneously a field called Higgs field was formed. I hope that is clear. Yes, I am using white Ishwar, but I am using the other colors when I want some contrast. Okay, I mean, like for example, I explained all these things here in the subatomic particles. So gluons I wanted to keep in a different color. In that situation, without the other option, I am doing it, right? Okay, now coming back to this. Okay, so soon after the Big Bang, there is no field called as Higgs field, but after some time the temperature cooled and a field called Higgs field formed. What is this? You know, electric field, okay, magnetic field, like that there is another field called Higgs field. Is it clear? There is another field called as Higgs field. Where is Higgs field? Here all, I mean, I am standing in the Higgs field. Is it clear? I am standing in the Higgs field. The earth is in the Higgs field. Sun is in the Higgs field. Stars are in the Higgs field. It is all pervasive. All pervasive. Present across the space. I hope it is clear. Right? Just like we have electric field, magnetic field, we have another field called as Higgs field. This Higgs field is all across the space. We have all the stars, planets, satellites, everything in the Higgs field. Like for example, I am right now standing in the Higgs field only. Okay. Every particle in our universe swims through the Higgs field. Every particle in the universe will swim through the Higgs field. Through this interaction, the particle will get mass. Very, very important point. Okay. So, this is a theory. Nobody knows how the matter is created. Nobody knows how these particles got mass. Something which have mass is called matter. So, how they attain the mass, how they are getting the weight, okay, that nobody knows. Okay, 2013 Nobel Prize in Physics was awarded to, okay, Peter Higgs and Englert for the theory on how the particles acquire the mass. So, nobody knows how the particles acquire the mass, but Higgs proposed a theory to explain how the particles acquire the mass. Higgs proposed a theory about how the particles acquire the mass. Okay, this is that theory, and that is the reason why it is called Higgs field, because this is proposed by Peter Higgs. Why this is called Higgs field? Because Peter Higgs proposed that there is one such field. Is it clear? Okay, I am going to tell you, Sadhana, what the Higgs field will do. I am going to tell you. Higgs field will give mass to the particles. Higgs field gives mass to the particles. I hope it is clear. Higgs field gives mass to the particles. How? Okay. See, all the particles are swimming through the Higgs field. These electrons, protons, neutrons, atoms, all these are swimming in the Higgs field. Any particle which will strongly interact with the Higgs field will be heavier, and any particle which weakly interacts with the Higgs field is lighter. All particles swim in the Higgs field. Why this is called Higgs field? Because Peter Higgs proposed this. And the particles that strongly interact with Higgs field are heavier. Okay, and the particles that weakly interact with the Higgs field are lighter, and the particles that do not interact with the Higgs field are, guess what? With the Higgs field are massless. They do not have mass. So what is the function of the Higgs field? Higgs field gives mass to the particles. Higgs field gives mass to the particles. I hope it is clear for all of you. Clear till now? Right. How does the Higgs field give mass? The particles that will interact strongly with the Higgs field are heavier. The particles that interact weakly with the Higgs field are lighter. And the particles that do not interact with Higgs field are the massless particles. That is how the Higgs field gives mass to the particles. But this is not a fact. This is only a theory. This is something which is proposed by Peter Higgs. Is it clear? This is just a theory which is proposed by Peter Higgs. Clear till now? And we have to prove this theory. We have to prove this theory. Is it clear? Okay? Right. Okay, we have to prove this theory. To prove this theory, they constructed the Large Hadron Collider. The Large Hadron Collider. Okay? Look at here. Like all fundamental fields, the Higgs field is also having an associated particle that is called Higgs boson. That is called Higgs boson. I mean, okay, if you compare Higgs field to an ocean, if the Higgs field is like an ocean, Higgs boson is like a wave. If the Higgs field is like an ocean, Higgs boson is like a wave. Is it clear? When do we get wave? When the Higgs field is disturbed, isn't it? If the Higgs field is disturbed, then we get the Higgs boson. Is it clear? Okay. How the Higgs boson particle comes? When the Higgs field is disturbed. Look at here. I tried to explain it here. Okay. In the Large Hadron Collider, that is an instrument. Okay. That is an instrument. This is called particle accelerator. This is called particle accelerator. So in this instrument, what they do is, okay, they send two protons. Can we call protons as hadrons? Just remember here, what are the hadrons we have seen? Hadrons are baryons, mesons. Baryons are protons and neutrons. So protons are a type of hadrons. Why this is called Large Hadron Collider? Because here two hadrons. What hadrons? Two protons are colliding. Okay, two hadrons are colliding. Therefore, it is called as Large Hadron Collider. Is it clear? Okay. So, in this instrument, when these two hadrons, that is when the two protons collide with each other, what happens? This collision creates disturbance. This particular collision creates disturbance in the Higgs field. I hope it is clear. The collision creates disturbance in the Higgs field, right? And yeah, when two protons are nothing but hadrons, two hadrons, that is when two protons collide with each other. In this instrument called Large Hadron Collider, this collision creates disturbance in the Higgs field. Okay, and as I told you, when the Higgs field is disturbed, okay, a particle will come out of it. That particle is called as Higgs boson. Okay, what is Higgs? Like I just compared Higgs boson to a wave and Higgs field as the ocean. When do we get wave? When the ocean is disturbed. Same way, when do we get Higgs boson? When the Higgs field is disturbed. how the Higgs field is disturbed when the two hadrons collide with each other. I hope it is clear. Two positive charges do not collide, Geeta. That is correct. That is the reason why we are using this billions of dollars, okay, instrument called particle accelerator. They are coming with very high speed. So they can, that kinetic energy can push the two protons together. They will fuse in this instrument. Outside they will not fuse. You are correct. I hope it is clear. Yes, Higgs boson is created. Higgs boson is created. Okay. No, electron is not a hadron. Remember, electron is a meson. Look at here. Electron is, just remember these words. If you forget these words, you cannot understand. Okay. Electron is a meson. Okay. Two hadrons, that is two protons collide. And Geeta's point is correct. Two protons, they both are positively charged. They repel each other. They will not collide. That is the reason why we constructed this Large Hadron Collider. Is it clear? Okay, that is the reason why $17 billion we are spending. $17 billion just to fuse two hadrons. I hope you understood this, Geeta. Right. Right. So, okay, now look at here. In the Large Hadron Collider, when two hadrons collide with each other, the Higgs field is disturbed. The Higgs field is disturbed and Higgs boson comes out, and it came out. They detected the Higgs boson and they call the Higgs boson as the God particle. They call the Higgs boson as the God particle. Why this is called God particle? Because it will create the mass. It will create the mass, or it will give mass to the particles. Right? Therefore, it is called as God particle. That is a fancy name, but it is called Higgs boson because Peter Higgs proposed this. I hope it is clear. Now let us read about this. Okay, Large Hadron Collider. A problem for many years has been that no experiment has observed the Higgs boson. Why should we observe the Higgs boson? Because when we observe the Higgs boson, okay, see, the presence of Higgs boson proves the presence of Higgs field, isn't it? I mean, wave proves the presence of the ocean. When there is no ocean, how can we get the wave? There is a wave means there is an ocean. Like that, when there is Higgs boson, definitely there is the Higgs field. Okay, so that is the reason why we are trying to detect this God particle or the Higgs boson. I hope it is clear. Okay, no Ranjita, electrons will not come under hadrons. Electrons will come under the lighter particles called mesons. I hope it is clear. Right? Then. Okay, and on July 4th, 2012, a new particle in the mass region of 125 giga electron volts was detected. This is the Higgs boson. In Atlas and CMS at CERN, okay, research center in Geneva, Switzerland, the Large Hadron Collider, okay. The Large Hadron Collider is having two equipment called Atlas and CMS. Okay, they collided the two protons. The boson, okay, was detected. A new particle in the mass range of 125 giga electron volts. What is this? This is the Higgs boson. This is the Higgs boson. Clear for all of you? No Rans, meson is not a hadron. Okay, just look at here one more time. Hadrons. Yeah. Yes, correct. Okay, it is a hadron. Yes, electron is also a hadron. Right, perfect. Okay, I was just thinking about the baryons. Electron is not a baryon, but electron is a hadron. Correct. Electron is a hadron. Look at this flow chart, it will clearly explain. Hadrons are two types, baryons, mesons. Okay. Mesons are two types, charged mesons, uncharged mesons. Charged mesons will form electrons and neutrinos. So electrons are charged mesons, and mesons are a type of hadrons. Yes, mesons are a type of, electrons are a type of hadrons. Right? Clear till now? Okay. Then, so which instrument detected the presence of the Higgs boson? The Atlas and CMS in the Large Hadron Collider detected the presence of the Higgs boson. Okay. No, neutrons cannot be used in the place of, yeah, neutrons also can be used, but they used protons. Okay. This particle is consistent with the Higgs boson. When two protons collide in the LHC, it is their constituent quarks and gluons that interact with one another. These high energy interactions can produce the Higgs boson. When the two hadrons collide with each other, these high energy interactions between the gluons, quarks and all will disturb the Higgs field, The disturbed Higgs field will generate the Higgs boson. Okay, that is about the Large Hadron Collider. And another news article, there are 3 news articles related to it. This is an important topic because there are total 3 news articles. Look at here, CERN proposed a collider 3 times bigger than the LHC. The other is about the Large Hadron Collider. The other one is about what is Higgs field. There are 3 news articles related to this over a span of some 2-3 months. Okay, and I kept all the 3 articles in one place for a continuity. I hope it is clear. Okay. Right. And what is this? Large Hadron Collider already detected the Higgs boson. Large Hadron Collider already detected the Higgs boson. Okay. But the CERN proposed a bigger collider than LHC. The CERN proposed a bigger collider than the LHC. Okay. A $17 billion future circular collider. This instrument will be like a circle. In this the particles will come, proton from here, high speed. Okay. And why do we need a larger collider? 3 times bigger. Because bigger it is, higher speed is possible. Bigger the collider, higher the speed possible. Bigger the collider, higher the speed possible. Is it clear? Right. Okay. This is 57 miles long. After all, subatomic particles called hadrons are moving 57 miles. Okay. When such a speed, like say for example, when you are bowling for the cricket also, you run and bowl. That is to catch the, like pick up the speed. So same way, when these protons are traveling for such a long distance, okay, they will acquire the kinetic energy, and when with such a high speed, when they collide with each other, they can fuse. Otherwise they cannot fuse because they both will repel each other. They both are positively charged. Is it clear? Okay. So same thing. Okay. But bigger one. Okay. This is bigger than, 3 times bigger than 6.5 miles long Large Hadron Collider, which is the older one. The older one that we discussed here is 6.5 miles long. But CERN proposed a bigger one, 3 times bigger one. By smashing the particles, by smashing the 2 protons together at even higher energies, the researchers hope to find unknown particles. Yes, then you may ask me, ma'am, in the previous one only, that is this 16.5 mile long Large Hadron Collider already detected the Higgs boson, then why this 3 times bigger collider? Okay, to detect any new particles that we did not even imagine till now. Peter Higgs proposed that there is something called boson, we detected it. But who knows, there can be more particles that nobody even imagined till now. So for the detection of such particles, we are planning a bigger collider. Is it clear? Okay. The researcher hoped to find unknown particles, the particles which we do not know may be identified in this experiment. and unknown forces also may be identified. Okay, and to discover why matter outweighs antimatter. Yes, you people are asking me a few questions. What is dark matter? What is dark matter? Dark matter is the matter about which we do not know. Dark matter is the matter about which we do not know. Okay, and what is matter? Okay, matter is the matter about which we know. I mean, like all our stars, planets, okay, satellites, all the living organisms, cells, atoms, okay, electrons, protons, neutrons, all these things comes under the matter. Is it clear? Okay, and what is antimatter? Antimatter. Antimatter means when matter collide with antimatter. Okay, when matter collide with antimatter, both will, okay, annihilate. Both will annihilate. I mean, both will cancel each other. Nothing remains. If matter binds to antimatter, matter, antimatter, both will cancel each other. Nothing remains. That is called antimatter. I hope it is clear. Right. Now tell me, what do you guess? In the universe we have more matter or more antimatter? In the universe, what do you think? Do you think we have more matter or more antimatter? Yes? No, Avinash, because if antimatter, see. If antimatter is more than matter, all that antimatter will cancel the matter. Matter should not be there, isn't it? Antimatter, matter will cancel each other. If antimatter is more, then all the antimatter will cancel out all the matter. Then matter should not be there. And should not be equal also. I mean, if antimatter and matter are equal to each other, then this universe do not exist. If antimatter and matter are equal to each other, universe do not exist. If antimatter is more, antimatter exists. If matter is more, matter exists. So now tell me, what is more in the universe? What is more in the universe? Matter is more in the universe. See, okay. This is our total universe. Is it clear? This is our total universe. 72% of our universe is dark energy, not dark matter, please. Okay, don't get confused between those two. Not dark matter, dark energy. Right? Okay. And dark matter is 23%. 23% dark matter. Atoms means matter. Matter is only 4.6%. It means all our stars, planets, satellites, okay, inside the planet the living organisms, inside that cells, inside the cells atoms, inside the atoms the protons, neutrons, the hadrons, all these things, all of them form only 4.6% of the universe. I hope it is clear. Okay, and this larger collider is also, the purpose of this larger collider is to identify the unknown particles, to identify the unknown forces, and also to discover why matter overweighs the antimatter. I mean, this is a wonder, right? Okay, how it is possible, how the matter is more than the antimatter. Just if the antimatter is more than matter, then we would not have existed. So who decided, that is what decided, how the matter is more than the antimatter? Okay, so we should find out all these mysteries. This particular, okay, $17 billion future collider, which is 3 times larger than the Large Hadron Collider, okay, is like to discover the unknown particles, unknown forces, and to know why the matter is more than the antimatter, and also to understand. Probe means to understand the nature of the dark matter, because we don't know that is dark matter, okay, and also to know the nature of dark energy. What is dark energy? To understand all these things, we have constructed this instrument. I hope it is clear. This instrument is constructed for that purpose, okay. Then I will stop this session here today, but a small recap of the hadrons, because a small confusion created, okay. Atomic structure, just one more time. This is the atom and this is the nucleus. Inside the nucleus we have protons and neutrons. Inside the protons we have quarks. Quarks are 2 types, up quarks, down quarks. Quarks are held together by gluons, okay, and electrons are orbiting around the nucleus. All these fundamental particles are called as hadrons. Hadrons are 2 types, baryons, mesons. Baryons are 2 types, protons, neutrons. Mesons are 2 types, charged mesons, uncharged mesons. Charged mesons, both of them, charged and uncharged mesons, are very unstable, okay. They will convert into the other particles within 1/100th of a millisecond. Charged meson will become electron or neutrino, and uncharged mesons will become the photons. Photons and neutrinos are not parts of the atom, they are present free, okay. This is about the fundamental particles. And Higgs field is an all pervasive field across the universe, okay, and all atoms, protons, hadrons, neutrinos, all these are in the Higgs field only. The disturbance in the Higgs field will create the Higgs boson. Higgs boson is like a wave when the Higgs field is called ocean. Why the Higgs field is called as Higgs field? Because Peter Higgs proposed it. How to prove that the Higgs field exists? By showing the Higgs boson. How to create the Higgs boson? By colliding 2 hadrons. Where to collide the 2 hadrons? In the Large Hadron Collider, okay. Then the Higgs field will disturb, Higgs boson will come. What is the function of Higgs field? To create mass or to give mass to the particles. How does the Higgs field give mass to the particles? All particles are swimming in the Higgs field. The particles that strongly interact with the Higgs field will be heavier, and the particles which weakly interact with the Higgs field are lighter. This is a quick summary of all this topic. That's all for today's session. These are a few important topics, okay, from the duration of January to May 2024. We will also cover May 2024 to May 2025, that is last 1 year current affairs, in another phase of the Conquer Prelims 2025. Thank you so much.